Hi guys, welcome back to another new video in Microbiological Insights, and today in this video we will be going to discuss about the most important topic, that is the cell division. So if you want to watch this videos in Tamil, please subscribe Microbiological Concepts. So let's move into the video. What is cell division? Cell division means a single cell can divide into daughter cells, and this cell division is an essential means of reproduction for all the living things. This cell division allows the organism to transfer their genetic material to their offspring. So why do the cells divide? The cells divide to perform the functions such as growth of the organism, and it can replace the damaged or old cells. A single cell can divide into two cells, then these two cells again divided into two new cells. So the single cell divided into two becomes 4, and 4 becomes 8, 8 becomes 16, 16 becomes 32. Like that it goes on. So it creates a new populations of cells, that is called as clone. So all the cells within a clone are genetically identical, and this cell division is an very essential part of growth of all the multicellular organisms. What are multicellular organisms? Those organisms that consist of more than one cell. And this cell division is also the basis of reproduction. What is mother cell? A cell that is about to divide is called as a mother cell. What are daughter cells? After cell division, the products of that division are called the daughter cells. Next we move on to the cell division in prokaryotes. When prokaryotic cells divide, the contents of the mother cell are more or less equally distributed between the daughter cells. So what happens during the cell division in prokaryotic cells? If a single cell divides, the content of the mother cell, so all the contents which are present within the mother cell are equally distributed to the daughter cells. This process is termed as fission. So what happens during this cell division? The mother cell's chromosome is duplicated prior to the fission. So before fission, the mother cell's chromosome is duplicated and the copies of the chromosome is transferred to each of the daughter cells. So they are equally distributed. Let's see with an example. Under optimal condition, a prokaryotic such as the intestinal bacterium E. coli. So you all know about the E. coli. Escherichia coli is an intestinal bacterium. So under optimal condition, this intestinal bacterium Escherichia coli divides every 20 to 30 minutes. If this rate continues, a single E. coli cell could form a clone of approximately 2 power 50 cells, that is more than a quadrillion cells. A single cell can produce more than a quadrillion cells in just one day. But this is not the reality. When we see the reality, these E. coli cells do not sustain this high rate of cell division. So what happens is, if the cells accumulate during the cell division, the rate of cell division declines because the nutrients present in that particular environment or the medium are exhausted. And another important thing is the waste products getting piled up. The waste products are accumulated in the medium, but the nutrients are exhausted from the medium. So a single E. coli cell can produce enough progeny in one day, not this much cells. And this single E. coli form a mass of cells. We termed as that mass of cells as a colony. Within the colony, all the daughter cells are similar, and these cells originate from a single mother cell. And now let's move on to the cell division in eukaryotes. The division of the eukaryotic cells is a more elaborate process than the division of prokaryotic cells. Prokaryotic cell is an unicellular organism, so the cell division is little faster when compared to the eukaryotic cells, because the eukaryotic cells are multicellular organisms and it is a elaborate process. What happens during the cell division in eukaryotes? Typically, many chromosomes must be duplicated and the duplicates must be equally and exactly distributed to the daughter cells. So, what happens to the chromosomes means the chromosomes must be duplicated and the duplicated chromosomes are equally and exactly transferred to the daughter cells. But the cell organelles like mitochondria, chloroplast, endoplasmic reticulum and Golgi complex are not evenly distributed to the daughter cells. So this process is not equal and exact. Only the chromosomes are equally and exactly transferred to the daughter cells, but not the cell organelles. What happens is mitochondria and chloroplast are randomly distributed to the daughter cells. So the mitochondria and chloroplast are cell organelles, no? So these organelles are randomly distributed. So it can distribute not in a systemic fashion. And the endoplasmic reticulum and the Golgi complex are fragmented at the time of division. So the endoplasmic reticulum and Golgi complex fragment at the time of cell division and it gets transferred to the daughter cells, and within the daughter cells it reformed into a new cell organelle. So this cell division takes more time and it is an elaborate process. Each time when a eukaryotic cell divides, it goes through a series of phases. So there are so many phases in the eukaryotic cell division. This series of phases we termed as the cell cycle. The progression of phases is denoted as G1 phase, S phase, G2 phase and M phase. So the S phase is the period in which the chromosomes are duplicated. So during the S phase only the chromosomes are duplicated and at that time DNA also synthesized. And during the M phase, what happens is the mother cell actually divides into a daughter cell. During the S phase, the chromosomes are duplicated. That is, the DNA synthesis happens. But in the M phase, only the mother cell actually divides into daughter cells, and this phase has two components. What are the components? The mitosis and cytokinesis. This is the overall cycle of an animal cell. This cycle is 24 hours long, but the duration of the cycle varies among the different types of the eukaryotic cells. G1 phase is too long, no? So it takes 10 hours for the growth and metabolism of that cell. And during the S phase, what happens? The DNA synthesis and chromosome duplication occurs, no? And after that is the G2 phase. The G2 phase is the preparatory phase for mitosis process. And this phase is the mitosis. M refers to the mitosis phase. These are the phases of mitosis cell division: cytokinesis, telophase, anaphase, metaphase and prophase. What is mitosis? Mitosis is the process that it distributes the duplicated chromosomes equally and exactly to the daughter cell. The chromosomes are equally and exactly distributed to the daughter cells, and this process is termed as the mitosis. And what is cytokinesis? The parent cell physically separates the two daughter cells from each other, no? And this process is the cytokinesis. M means mitosis. The name mitosis is derived from a Greek word. The meaning of that word is thread. And during mitosis, the chromosomes appear as thread-like bodies inside cells. G1 and G2 phases are the gaps between the S and M phases. And the length of the cell cycle varies among different types of cells. When we see the embryo, the growth is very fast and the cell cycle may be as short as 30 minutes. But in the slow growing adult tissue means it may take several months to divide from a single cell to daughter cell, no? And some cells such as the nerve and muscle tissues stop to divide once they acquire their specialized functions. If they reach that particular size and growth, after that the cell division gets stopped. And the progression of the eukaryotic cells through their cell cycle is controlled by different types of protein. When the protein function is disrupted, the cells divide in an unregulated fashion. If there is any problem with the protein, the cells cannot divide in an even manner. It divides in an unregulated fashion. This deregulation of the cell division may lead to cancer. So what is cancer? The abnormal growth of a cell, no? That leads to cancer, and this is the major cause of death among people today. These are all about the cell division, and this is the reference book. So if you have any doubts, you please write your doubts in the comment section. Okay, if you like this video, you can share it with your friends. Okay, thank you.
If you like this video, please don't forget to subscribe, share, like and click the bell button. If you have any more doubts, you can write your doubts in the comment section below. Thank you.